Hi, my name is Aneesh Kadambi, and currently I'm a senior in the MSCC program at Paul Laurence Dunbar High School. Today I'll be talking to you about my project, Amylin Dyshomeostasis, the link between diabetes and Alzheimer's disease. Now, I worked under the mentorship of Dr. Florin Despa at the Department of Pharmacology and Nutritional Sciences in the University of Kentucky. Now, before I begin my presentation, I'd like to begin with a brief overview of my talking points for the rest of the PowerPoint. So first, I'd like to start with my introduction, followed by my purpose and hypothesis. Then I'll get into my methodology, my results, and my discussion. And lastly, I'll finish up with further research in this field and my acknowledgements and my references slide. So first, a little bit about Alzheimer's disease as a whole. As I'm sure you guys know, Alzheimer's disease is an irreversible progressive brain disorder, and this slowly destroys memory and thinking skills, and eventually the ability to carry out everyday simple tasks. Now, right now, Alzheimer's disease pathogenesis is widely believed to be driven by the pathological production and deposition of the beta amyloid peptide. Now, what is the beta amyloid peptide? These are the main components of the amyloid plaques found in the brains of people with Alzheimer's disease. These peptides derive from the amyloid precursor protein, also known as APP. Now, these amyloid beta molecules can aggregate and eventually form oligomers, which are toxic to nerve cells. According to the amyloid hypothesis, these stages of beta amyloid aggregation eventually disrupt cell-to-cell communication and start activating immune cells. Now, these immune cells will eventually start to trigger inflammation along the blood-brain barrier. This is widely believed to be why the blood-brain barrier is involved in the pathogenesis of Alzheimer's disease. The blood-brain barrier is a highly selective, semi-permeable structure and chemical barrier. Now, this ensures that we have a stable and internal environment of our brain and prevents foreign objects from invading the brain tissue. A progressive accumulation of beta amyloid in the brain and blood-brain barrier dysfunction may give rise to cognitive impairment and eventually the onset of dementia. Now, a blood-brain barrier dysfunction will start to trigger neuroinflammation and oxidative stress. One of the most common risk factors then associated with sporadic AD patients is diabetes. And in a lot of cases, some research groups actually call Alzheimer's disease type 3 diabetes. Amylin is a peptide hormone that is co-secreted with insulin from pancreatic beta cells. In pre-diabetic and type 2 diabetic conditions, hyperamylinemia predisposes to aggregate and form these kind of amyloid oligomers and fibers, and these may play a part in the damage done to the blood-brain barrier and dementia. As you can see in this picture, the blood-brain barrier is shown with all of its components. The endothelial cells line the tight junction and provide a selective transport of blood and biomolecules into the brain. Now, there are also cerebrovascular risk factors, also known as CVRFs, which induce platelets and platelet activation, resulting in increased vascular inflammation and increased endothelial cell permeability. Now, these activated platelets eventually secrete amyloid precursor protein, also known as APP, which is then cleaved to cytotoxic amyloid beta peptides. These alterations in blood-brain barrier function can lead to an increased amyloid beta uptake and amyloid plaque formation in the brain. For my purpose, it was to prove human amylin oligomers disrupt blood-brain barrier function. And my independent variable was expression of the human islet amyloid polypeptide treatment versus the vehicle control. And my dependent variable was different gene expressions in the blood-brain barrier function and integrity, and animal behavior. My hypothesis was that human amylin oligomers aggregates disrupt the blood-brain barrier function. For my methodology, an in vitro model was used for the rat brain microvascular endothelial cells to demonstrate how amylin oligomers affect blood-brain barrier function. These RBVEC cells were treated with human islet amyloid polypeptide, oligomers, and the vehicle control as well. For this, Western blot and immunofluorescence were done for different cell signaling protein molecules involved in the function of the blood-brain barrier. For the in vivo model, human islet amyloid polypeptide overexpressing rat mouse models were used additionally to demonstrate the HIAPP damage for the blood-brain barrier. These animals were then bred and genotyped, and some of the organs, like the brain and pancreas, were collected and frozen for further experiments. PCR was mainly used for genotyping and for additional cell signaling proteins involved in blood-brain barrier damage was done as well. Now, for the Western blot, the RBVE cell lysates were used, and they were the protein concentration was also quantified by a BCA. After this, the PVDF membrane was incubated with the primary antibody, which was the Z01 polyclonal antibody, and the secondary antibody, horseradish peroxidase, or also known as HRP. GAPDH was used as a loading control, and after washing the membrane using a G-box for imaging, the image of the membrane was then taken. For the immunofluorescence, RBVE cells were also used and treated with the HIAPP peptide or the vehicle control before being fixed with paraformaldehyde. Now, these fixed cells were labeled with the Z01 antibody as well and detected with the Alexa Fluor 488 IG under a fluorescence microscope. Now, for this, as you can see in the picture, these antibodies were tagged with the fluorescence and they were combined with some sort of fluorochrome, as you can see in the picture. After this, these were bonded with the antigens on the cell surface so that they can be combined with the fluorescence and along with the antibodies as well. For PCR, genomic DNA was isolated from mouse tail clippings. And this was used as a template to amplify the gene of interest along with the PCR master mix and the forward and reverse primer set. Now, after amplification, these cells were mixed with loading and visualizing dye, and after this, they were loaded into a gel with the DNA marker, positive and negative controls, and a picture was taken after the gel was run through electrophoresis. As you can see in this diagram, this is the repeated cycles for when the samples were loaded into the PCR machine. And as you can see, they are repeated 40 times for 40 cycles and vary between the temperatures in each 6 steps. Now, to get on to the results from the Western blot, as you can see in the vehicle control, compared to the HIAPP treated endothelial cells, the Z01 tight junction protein showed significant reduction in both size and integrity compared to the vehicle control. And in this case, GAPDH was used as a loading control as well. And it was not affected for both the vehicle control and the HIAPP treated endothelial cells. So we can say that the results were kind of verified by this loading control because it was seen to be not affected by either treatment. Along with this, the immunofluorescence imaging showed a significant reduction in overall Z01 tight junction protein in cells treated with HIAPP versus the vehicle control. As you can see in this picture, with the vehicle control, the Z01 tight junction protein is seen to be mostly encasing all the nuclei and intact and put together, compared to the HIAP treated cells, where most of the integrity is now gone and the nuclei are not surrounded with the strong Z01 tight junction protein anymore. Additionally, for the interleukin 1 beta, as you can see with the amylin treated, with the human islet amyloid polypeptide treated cells, compared to the vehicle control, there was a significant increased synthesis of the interleukin 1 beta in the rats expressing the human amylin compared to the vehicle control, with the APOE3 allele as well. Additionally, for the results for PCR, the APOE3 HIAP overexpressing mice were genotyped based on the wild type, homozygous, and heterozygous positive controls successfully. So, the results of the experiment show that the HIAPP treatment in brain microvascular endothelial cells decreased tight junction protein Z01 overall, which means my hypothesis was supported that overall hyperamylinemia would decrease the blood-brain barrier function. This decrease in the tight junction protein weakens the kind of regulation of the non-specific influx and efflux of all the biological molecules across the blood-brain barrier, signaling a kind of disruption of the BBB overall. Additionally, the HIAP treatment increases the synthesis of interleukin-1 beta, which suggests increased inflammation and neurodegeneration along the blood-brain barrier. Specifically, IL-1B increased astrocytic production of pro-inflammatory chemokines, and since the blood-brain barrier is formed by endothelial cells, these results suggest that the function of the blood-brain barrier may eventually get compromised by this oligomerized amylin. However, a statistical test for some sources of error would be needed to conduct to verify further results if the to actually know if the results were significant. Overall, this supports some previous research done on the role that amylin contributes to the progression of Alzheimer's disease. In addition, the studies show evidence of these molecular changes as kind of small changes that will eventually lead to a cascade of full biological changes that might lead to a systemic wider disease. These studies are important to consider in this field of Alzheimer's because the understanding of the whole pathology of these diseases is necessary before even establishing a link between diabetes and Alzheimer's disease. Now, some potential errors other than in actually setting up the experimentation would include setting up cell lysates, which could have led to variability in Western blot and immunofluorescence results. More rounds of the Western blot and immunofluorescence could limit maybe some of the possibility of human error and increase the reliability of the results. Now, for further research, further experimentation definitely involves screening additional gene expression levels, such as the apoprotein APOE gene. We could additionally screen for the APOE2 and the APOE4 as well. For example, this could possibly be done by somehow making a knockout model and then potentially checking the blood-brain barrier integrity by in vitro and the in vivo model. Additionally, the role of other cells in the blood-brain barrier could be investigated, such as the function of astrocytes in regulating tight junction protein. Additionally, future studies could involve observing some of the longer-term effects of the oligomerized amylin on the blood-brain barrier, since many of the biological changes in Alzheimer's disease occur later in its progression. Although Alzheimer's disease has continued to be researched currently, there's still a lot to be explored for developing an effective link between diabetes and Alzheimer's disease. For my acknowledgements, I'd like to heavily thank Dr. Florin Despot and Dr. Vel Murugan Viswathan and the whole Despot lab for allowing me to come. I'd like to thank the Kentucky Young Researchers Program. I'd like to thank Paul Lawrence Dunbar, the MSCC program, and especially Ms. Karen Young for giving us the opportunity to carry out this research. And lastly, I'd like to thank family and friends for helping me along the project, for helping me along the way, and helping me with my project. And lastly, I have my references slide. I thank you for listening to my presentation. And this is Aneesh Kadambi signing off.